stimuli are occurring, this is going to continue on and continue on and continue on. So it's not by the negative feedback loop that we did before. Same thing with the other one. When we did PIH, you know, you have this idea that we're decreasing PIH, okay? Therefore, we're increasing prolactin. Again, this is under the same stimuli. And then prolactin, and then therefore increasing milk production. And again, no, this is not causing negative feedback on that, as far as I know, right? It's basically, as long as the stimuli is occurring, we're going to keep doing this so our milk production stays up as long as it's needed.
So do those happen simultaneously?
So,
because again, if you look at it, because I guess they're not showing the pathway down for the higher brain centers, but if you just look at this, it's a simple mechanoreceptor causing an afferent that sort of, again, branches off in the hypothalamus to do both of those things. And again, nervous system, remember, is extremely fast. So, yeah, it's basically simultaneous.
so what kind of stimuli would inhibit PIH?
What kind of stimuli would inhibit? Well, this is inhibiting the PIH.
Oh, well, yeah, what kind of stimuli? Is there a specific? Just, okay.
As far as I know, child crying, nipple suckling. Say that 3 times real fast.
Oxytocin and this one.
Yes.
Because they happen simultaneously.
Right. I don't know if I'm playing a lot with that. Okay, but that's that.
Did we do a positive, well, we didn't do because it was already up, a positive feedback loop for childbirth? I mean, it was the second to last slide.
I mean, if you want, if you were trying to do it like this.
Would we need one, really?
Not really. I mean, these positive feedback ones, you have to just know them. There's not a lot of manipulation. I mean, I'm not going to have, some sort of weird tumor while she's having a baby or something like that. Even my mind doesn't really go that much. So it's the only one, really it's sort of knowing what's happening here, okay? Understanding the system. I don't know if I'm messing this up too much. I have to look at my test to actually determine that. And this one, again, understanding the hormones during ovulation, basically, versus not, okay? So it's sort of factual in a way, more than trying to really manipulate. The ones we manipulate are all these other ones when we have, the scenarios that we talked about, the other 4. So you could do the male one because it's a lot simpler, but you could manipulate the female one a little bit too, I guess, yeah.
So the 3 positive feedback loops associated with female ovulation, childbirth, and breastfeeding, what are the external factors
That stop which one?
that stop all for
Well, ovulation it is, the ovum coming out, so the ovulation process, right? The follicle bursting and doing this. That stops the process. For this, it's the child being born. Okay, we're not stretching the cervix anymore. It's done. And then I don't know about this. Then I don't know, just not doing it for a while. I don't know exactly what stops that. I guess it's that. Yeah, I'm a little phased on all the details. What?
Did you ask your wife?
I don't think so. I don't know if she'll know that. It's been a while. We're pretty old, my son's 24. It's been a long time since she breastfed.
I'm sure she still remembers.
What?
I'm sure she still remembers.
Okay, I love it in the course. And you guys have been really good, okay? This is like the most I've been harassed on breastfeeding since I don't remember when, okay? And clearly you can see what I know. Okay. What do I need to do? First of all, you really want, you need to look at those questions. You need to look at those questions. So look at the questions and see which ones I sort of want, especially I don't want to just do the multiple choice because they're already there, right? You have the answers if you just want that. do you, first of all, what questions overall do you have, and can you work through those multiple choice? If you don't have the multiple choice with you, go to someone who does, so you can actually start looking at those. Yes?
Are you gonna give us any kind of hint as to what the essay question?
There is no essay. All multiple choice.
Oh.
50 multiple choice. That way I can get it back and have your grades in right away. So 50 multiple choice. So that's an indication what the essay is. So that's good in a way, because you don't have to worry about some thing. And again, all these terms, abbreviations are simply going to be on the test, and I don't think there'll be a problem. You'll know who's who, so to speak. Well, she had a question for us.
Just can you explain the thyroid, like how kind of the receptors work down in T3 and T4?
The
I'm still a little confused about how hypo and hyperthyroidism can cause goiters.
Okay, so the real key to answer both those questions, okay, we're talking about, let's go through again, this is it's not the only slide that you can use, okay, because you can do, again, tumor, destruction. Those are the other 6 scenarios. These are 2 more. But when we're talking about the goiter aspect, okay, we're talking about the goiter aspect, the reason this is good to illustrate is that we're going to see it can be hypo or hyper, because that's referring to the level of the thyroid hormones, right? If you don't have enough, you're hypothyroid. If you're too much, you're hyper. Both of these conditions can cause goiters. The goiter is not these hormones. It's this stimulation, okay? So, again, you have, let's get to the anterior pituitary. And again, I have to emphasize for all these, remember, for all these except for oxytocin and ADH, we're talking about the anterior pituitary. And some of my answers, A, B, C, D, I'm going to have disruption of the posterior pituitary or something like that. And so if you thought it was disruption of the pituitary in one of these systems here, don't put posterior pituitary unless I talk oxytocin. So make sure you know how you make, again, the real thing with multiple choice that a lot of you that are really smart sort of make mistakes on are sort of like simple mistakes. Like decrease versus increase, or anterior versus posterior. You remember, on my test you have unlimited time. Take your time. Anything you think you might make a mistake, or that, that again, people will call those tricky questions, whatever you want to call it. I call them, carefully looking over the questions, okay? Critically thinking the questions, is, underline these words, okay? Like anterior versus posterior, to make sure you're not going to make that mistake. So anything like that, those little, if you want to call it tricky, the things that can trick you up, to underline the words or highlight them like that, so that you don't make it. So make sure we're dealing with anterior pituitary. In this case, again, we have TRH, TSH, and when we're talking about the thyroid, we're talking about the follicular cells. Remember, the C cells are for calcitonin, calcium balance. It's the follicular cells, and that's why the real key is, on these follicular cells, too bad that other picture didn't show it, if we look at kind of the follicular cells here, that's doing all this process, there are TSH receptors. And remember, when we're going down this way, TRH stimulates the release of TSH. TSH will stimulate these follicular cells normally to make T3 and T4. And then you'll have the negative feedback. So this is normal. But for the goiter aspect, so this will be the same way to have hyper or hypothyroidism, right? You can play the same game, tumor, destruction. But when we're talking about the actual goiter, if you have a goiter, you have an enlarged thyroid, it doesn't matter on the levels of the hormones. It's these follicular cells that, again, if you look at them too, even from this picture, they have this sort of, glycoprotein, sort of central lumen that's involved in a lot. And if you start stimulating these cells, like with TSH, you basically are going to cause these to get larger and larger. It doesn't matter if you're making T3 or T4. It's this. It's the stimulation of those cells to try to do all that process. Okay? So, to go through all these steps to make it. So, if you look at the examples, let's see if this will make sense then. So, you can have, so that's the key. Everyone just got to realize it's this process. It's the stimulation of these TH receptors on the follicular cells that are causing the goiter, causing the enlargement of the thyroid. It's independent of the hormones being produced. It's this stimulation. It's TSH, or in one case we're going to see an exception. Okay, it's going to be TSIs, which act as a TSH agonist. It's acting like TSH.
So that's still producing the T3 and T4, the
Yes.
TSI.
Yes.
Okay.
Okay, because you got plenty of iodine. In this case, if you can, this is normally TSH is causing these follicular cells to make T3 and T4. Okay, but in some cases, even if they don't make it, you're still going to stimulate. See, here we're stimulating those cells with TSH. Here we're stimulating those cells with TSI. It is that stimulation that's causing the goiter. It's excessive stimulation of these follicular cells. Either TSH directly, which is normally what happens, or in this one scenario, we have that if you have Graves' disease, you're making antibodies that in this case actually activate those cells. Okay, they act like TSH. They're TSH agonists. That's the key element here. You're looking for a goiter. TSH either must be high, or you have Graves' disease. Those are the scenarios you're looking for on the test as far as what could cause a goiter. The key is, it doesn't matter what these other ones are, but you're looking for TSH, or Graves' disease. If you just follow these two scenarios, you can see the different levels. So, again, without iodine, you don't have T3, T4 because you need iodine. Therefore, you're going to try to make it. TRH and TSH is up. The key with the goiter is this. Okay, TSH is going to stimulate these follicular cells to make it. Without iodine, can't make it. So we're not going to have any negative feedback. That means we're going to keep making more TRH, therefore more TSH. This is going to hit this. Say make it, can't make it. So it is what's causing the goiter here. We have low T3, T4. We have hypothyroidism. You're going to have all the symptoms of a low thyroid activity. But you're going to keep hitting this with TSH. That's causing the goiter. So it's not the hormone itself, it's the stimulation. It's the stimulation of these receptors, these TH receptors on the follicular cells.
So the goiter is actually caused by the lack of negative feedback.
In this case,
Right.
exactly. Okay? So the lack of negative feedback means that TSH levels are going to be really high. If you have, what's another way? Well, let's just do this way. And then we can look at it.
So wait, so you have high TRH, high TSH, and low T3, T4?
Yes.
Okay,
But you still have a goiter because
Yeah.
of this right here.
yeah. But that's what we expect to see in the clinical.
Right. In this case, you have this idea of sort of the opposite way here. We're going to have your Graves' disease. You've got to realize you're making antibodies that will act like TSH. Okay? They're going to bind to those TH receptors, activate these cells to make T3, T4. And they're going to make T3, T4 because we have iodine. Now, because of that, again, it says here, strong negative feedback. Now you expect these guys to be the opposite because of the feedback. We're making all this. You're going to say stop it, so you're going to have lower levels of TRH and therefore lower levels of TSH. So here you're like, okay, I'm not stimulating that. It should be okay. But the TSIs do not respond to that feedback. Okay? So they're going to keep hitting this and keep stimulating that so that that's what's causing the goiter. Okay? It's overstimulation of these receptors. Either TSH itself or a TSH agonist. Remember, TSH, an agonist operates the same way. It's binding and causing the same effect. Okay? And again, it's not in control, basically. I mean, it's not being controlled by negative feedback.
So if you put TSI, will you put TSI ever?
What do you
know
mean
what I'm
put
saying?
it?
Like, for exam questions, will you give us an option being like, when you do up arrows or down arrows or whatever, you give us the symptomology, will you put TSI as one of the symptomologies?
I could, or I could just say Graves' disease. And you have to know it's high TSIs.
Okay, exactly.
But if it's Graves' disease, you've got to know this. Okay, this scenario, if I just say Graves' disease, you've got to realize these are the levels of the hormones. So you have to sort of get that or memorize and understand it's TSIs.
Did you just describe Graves' disease as an autoimmune? I know that you differentiated saying that it usually means destroying the tissues, but in this case it's stimulating them. But that's something like you're born with or you develop or?
It's genetic.
So genetic, so you'll have it like your whole life?
Yeah, although again, a lot of these autoimmune disease are, the expression of the genes are progressive, but it is during childhood, and then young adults, sometimes they really move up. Crohn's disease is another example of that. So genetically you have it, but when you actually have symptomology, it kind of varies, over the period of time. I don't know when this usually hits most people, but it's sort of a common one. Sometimes I have people in the class that know somebody with Graves' disease, so it's a relatively common autoimmune.
And the TSIs are the antibodies.
Yes. So they're the antibodies that usually destroy things. Again, when you talk about Crohn's disease or rheumatoid arthritis or type 1 diabetes, you're making immunoglobulins, antibodies that are attacking your own tissue and destroying it. I don't know why this is this way, but this one is actually opposite, where it actually stimulates the actual tissue to cause, again, it's still a problem, but it's what it's doing.
So the normal levels for the TRH, TSH, and T3, T4 would all be high, right? Or no?
Normal, this is where it's confusing with the arrow type thing. Normally, if the idea is...
It'd normally be high,
No,
and
no,
then it would be low, right?
No, it's just like, that's why, the arrows are saying, that something is higher or lower than normal.
Okay, but normally you'd have TRH stimulates TSH, creates T3, T4, which then stops the whole thing.
Yes.
Okay.
And then you're within that homeostatic range,
Okay.
exactly.
So that's...
And that's, because sometimes the arrows are confusing, because it's like,
Yeah, but so normally it's just like a flow chart, so that's why you left it with just the arrows in the middle, because it's saying, that happens to that, happens to that, and then it stops.
And then, it turns it down, it comes back to normal. So when we're doing these arrows like this, we're
So
saying
it's just for
they're
things that-
higher or lower than normal. We can measure those in the
Okay.
blood versus what the normal range of those hormones should be. And that's how we can start differentiating where the problems are lying. That's sort of the whole point of this class. Someone else had a question before?
I have a question. I wanted to go over question 15.
Which is?
Untreated type 1 diabetes often leads to all of the following symptoms except, and then it has hypokalemia, dehydration, glucosuria, metabolic acidosis, and ketone production.
Yes. Okay? So if you're trying to answer that question, again, two things. Okay? You want, first of all, and not a bad thing to do, because you're going to need for other questions, as I'm like doing this, is your blood glucose. So while we're doing that, let's just review that. Okay? If you are hypoglycemic, again, what hormone do you produce? If you have higher blood sugar, you're producing insulin, and you already know the cell types. Okay, remember, for both the insulin and the glucagon, we're talking about kind of what they're doing to the sugar, but they're affecting overall metabolism. And you remember all those names for synthesis and degradation of your proteins, fats, and sugars when you're doing this. And it's affecting all of it. Okay, so let's just review this because it's good for a lot of questions besides that, and then we'll kind of get to the diabetes. So high blood sugar, insulin, what is insulin going to do?
Increase glucose.
So we have glucose out here, and we want it into the cell. Okay? So normally we need what? Glut transporters. But insulin is going to have to be needed for most tissues to bind to its receptor. So again, insulin is going to, when we want to get glucose in, we have high blood glucose, it binds to its receptor, activates these glut transporters so that glucose can get in through the transporters. Okay, that's what we're doing with insulin. Okay? So I just want to put that, just to have this, increased glucose entry. Okay, the second one was that glucose, we put it in the cell to use it for energy. and make ATP and get CO2, we breathe it out. All those steps of aerobic respiration, and and basically insulin promotes that. So insulin promotes glucose breakdown. And I know it's a ways from your question, but again, you need to know this, what's involving it. Okay, 3, 4, and 5. So what are 3, 4, and 5? We basically got the idea of its main thing of getting glucose in, and we'll have the negative feedback. But that's not all insulin's doing. What else does it do? Anabolic reaction. Okay? So protein synthesis. Fine. What else? Lipogenesis. Insulin promotes fat. Okay? You think about diabetics having high insulin for type 2. So it's going to be protein synthesis. Lipogenesis. And we're going to store glycogen, glycogenesis. Okay? On the other hand, glucagon is going to promote, okay, we have low blood sugar and we need it, so it's going to promote glycogenolysis. What else is it going to do? Okay, now we're going to break down everything else to save sugar for the brain. So proteolysis, lipolysis. But we have sort of those extra features, don't we, to help us and survive. We have gluconeogenesis. And we have ketogenesis, which is converting fatty acids to ketones, emergency fuel for the brain. Remember, gluconeogenesis was making glucose out of non-carbohydrates, okay? Some of these breakdown products. By the way, besides glucagon, what other thing have we just talked about that responds to hypoglycemia and is needed for glucagon action and does all these things? So cortisol. So don't forget that. It's directly involved in all these processes. That's really important. So, for type 2 diabetes, actually, while we're doing this again, we have type 1 and type 2. Okay, for type 1 diabetes, what is it? So it's an autoimmune disease, so you have low levels of insulin because you're destroying your beta islet cells, and so these are insulin dependent. Okay, and for type 2, especially the early stages, what is the issue here? Your cells are becoming or are insulin resistant at different levels. So that your insulin receptors, and a lot of it has to do again with what fat releases, are not responding to the insulin. And so, what are your insulin levels going to be? Low or high? In response to it not working, you're actually going to produce high, at least at the early stages, high levels of insulin. Okay? Which is going to promote, again, all those processes, including lipogenesis. Okay? So diabetics, which a lot of times the obesity causes the insulin resistance, it's also by having a lot of insulin, you're going to actually promote more and more fat production. Again, it's sort of, you can spiral out of control. Now, to further answer your question, okay, to actually get it, but again, while we're reviewing, you need to know all this stuff for other questions. When you're talking about diabetes, you have high blood glucose, but glucose is not getting into your cells effectively. Your brain thinks you are starving, okay? It thinks you're not getting enough glucose because by all its measures here, it's not getting in the cells, so therefore you probably don't have it. It's going to react like that, okay? And so one major sign of uncontrolled diabetes is ketones, ketoacidosis. Okay, so diabetics, the reason they're producing ketones is, again, that their body, even though they have high blood sugar, because it's not getting in, their body is basically assuming that they're starving. And so they're going to start doing the emergency fuel for the brain, which is ketones. And again, ketones form that basically become acidic, which is what? Ketoacidosis, which is a type of metabolic acidosis. Okay? So if you look at this, okay, so the question is, which one doesn't apply? Okay? And again, you have A was what? Hypokalemia. We'll get to that. So what was B? Dehydration. Is dehydration true? Okay? And we did that last section. So we're looking for a false one. Well, this is true, so we do that out. We'll come back to that. C is glucose in the urine consistent?
Yes.
Okay, and so everyone do that. Okay, they do that. Now we're on to A, D, and E. So D was metabolic acidosis. And the other one was what? Ketogenesis or ketone production.
True and true.
So now we know, and I said it before, but again, that's why I put this on so that we can make sure for the people that care that we're emphasizing something that maybe I said, but it's easy for me to say it versus didn't really sink in at that point. So that's a big feature of it. Okay, ketone production and therefore metabolic acidosis. Okay, and so already you probably could eliminate, or you probably could identify hypokalemia as the one that's false. But you're like, well, what if not? Is that really true? Or did you make a mistake, Mike? So, and of course, I make a lot of mistakes. But where is this going? So there's a little, it's not a full cumulative type thing, but you know how I am. This is metabolic acidosis. What responds to acidosis in the kidney? Type A. Okay, responding to acidosis. We are absorbing bicarbonate to neutralize the acid. We're getting rid of the hydrogen. What else are we absorbing? So, as a response to acidosis, would we have hypokalemia? Hyperkalemia. If I wrote hyperkalemia, all of them would be true. So, again, that's why this is the one that's false. It should be hyperkalemia. But if you weren't sure about that, you need to be sure about all this, and you could probably eliminate that as well, okay? But you had to know, you had to remember this idea of that even though you have high, glucose levels, you basically are going to, diabetics are going to have ketone production and, uncontrolled and acidosis. So all those are consistent with it.
Would the principal cells react too? Because that's how I did it, or is that?
But then if they're going to do that to try to correct it, then it's going to affect blood pressure. I didn't even get to that. But these, it's going to be the primary response is to the acidosis. Okay? And remember how we did this thing. It's like at the very least, if the principal cells respond to that now, all they're going to do, we didn't make them get hypokalemia, right? We said maybe it would correct that condition, and then you'd have to get to the high blood pressure one. And I'm not going to actually go that much further on that. So, but this would be the immediate response, right? Because it's responding to the acidosis from this.
But isn't that how you know that it was dehydration? Is because it had to be the principal cells?
Dehydration is not the principal cells. Dehydration is what? What's causing the dehydration?
Glucose in the urine.
The glucose in the urine. So that's the
It's
directive.
Like pulling the water
It's
out.
pulling the water with it, right?
That's actually why I did principal cells, not in reaction to A, but because the glucose and the water would leave, which would lower blood pressure. So would the principal cell react by the blood pressure?
It might, yeah. But it's not going to, you're not going to have hypokalemia.
Right.
Well, if you were, if you were just dealing with that and you were talking about an overreaction of the principal cells, you could see how it could be hypokalemia then, okay? But that's not what I have here. It's really responding to the metabolic acidosis. And again, even if you could, I'm not saying you can reason that way, but then all these are true. And that's the idea too. It's like, unless you didn't know this, if you've got things which you know, and then you're not sure about, and you can even reason both ways, go ahead and pick the one like, that's that versus this. Because none of those are wrong, basically. And so that's sort of it. I understand what you're saying though.
Is there going to be a lot of questions that draw back from the previous?
Just a few. Okay, but again, what do I have on there? I have probably on the extra questions, including this one. So that's why I put these extra questions, because again, you are drawing back. We talked about Addison's disease on there, and we talk about how that affects aldosterone, which affects blood pressure, okay? So that's another one, but it's on this extra question right there. Besides that, no. Yeah.
Okay, so that was my question, just how much we should carry on from last chapter, how much we should review from last.
Again, besides this, not much of this, but again, the only other thing is aldosterone, okay? So when you're talking about Addison's disease, you're talking about destruction of the adrenal cortex, and we're mainly talking about cortisol, but we are talking about aldosterone, okay? And so if you have Addison's disease, and again, we can even do, let's actually look a second at Addison's disease, okay? So we have hypothalamus, we have the anterior pituitary, and we have the adrenal cortex. Okay, and we have what? Hypothalamus makes CRH. It's always going to be, I know it's a dyslexic thing, it's always going to be releasing hormones. Okay, anterior pituitary. And again, we're mainly talking about in this loop cortisol. And of course, we have that control loop. Okay, so let's just get the basics here, and then we're going to apply a couple things with this. So, cortisol. Okay, so what is Addison's disease? Destruction of the adrenal cortex. So right over here, we're going to say cortisol is okay, does everybody have that basic, right? That you have low this, therefore less negative feedback, you'll have high and high. Okay, so that's level. So, or that's fine. So we have low cortisol. Okay, but of course we also know we have low what? If you're destroying the whole cortex.
Low ALD.
Low aldosterone, exactly. So, again, now that one we do need to know, and I have it on there. So let's just look at what that means. And so, I'm not doing a lot of symptomology of this, but since we've just done aldosterone for blood pressure, and since we are doing sugar levels, this is going to affect both of those. And let's see if we see what that will do. So for blood pressure, how is that going to affect blood pressure? Okay, well, what do we have? Which one is involved in blood pressure over this? Aldosterone. And so this one you do have to know. If you have low aldosterone, okay, again, we have the principal cells now. Normally what? Sodium, potassium, excretion. Okay, low aldosterone means what? So we are going to basically lose or secrete sodium. Of course, you are going to reabsorb potassium and H2O, and therefore blood pressure. So with Addison's, you do expect a lower blood pressure. Okay? Maybe a little hyperkalemia, but I probably won't go there. Okay? There's that. Okay? How about, what do you expect sugar levels to be? High or low?
If cortisol is down there and you have low,
Do you have cortisol or not?
you do not.
What is cortisol used for? To do what to sugar levels? So, are we going to be able to do that? So we're going to have chronic hypoglycemia. Okay? So that's applying it to two things that we did. So as far as going back to the kidney, it is sort of knowing this idea of aldosterone with blood pressure with the adrenal cortex, and only the idea of metabolic acidosis. That question's like on the test. Like, you'll see that question. Okay? So actually, I think both these questions will be on the test.
Why do you have secrete sodium, and then do you have sodium and water? Why do you have the H? Is that why?
Yeah.
Right.
Yeah. I mean, when you reverse this, sodium's going to be excreted and water will follow, and that's why you'll lower blood pressure when you lower aldosterone.
So is the opposite also true if the cortisol is up? Does aldosterone also go up, or that doesn't affect
It can, but I probably won't do that. So it depends. There's a little variation of that, but basically, yeah. So if you have hypercortisolism, if you have Cushing disease, you are going to have sort of overactivity of aldosterone as well, and cause the opposite effect.
What you were saying over here is that if you have low cortisol, then it can't help glucagon to raise.
And
blood sugar.
it not only does it help it does some of the things itself as well. So if you look at the bullet points of cortisol, it responds to hypoglycemia, okay, just like glucagon. It's necessary for that activity, and it also promotes, these basic processes as well, directly too. So indirectly and directly, right, it's not going to be able to do that, and you won't be able to return sugar levels up to normal with that, and that's a major problem.
I think diabetes throws me off, like, in how it fits in here, because I know that, like, it's a destruction of the beta islet, which doesn't allow you to produce insulin.
Well, the type 1, right. Okay.
Yes, type 1. So when you had originally talked about that question about ketogenesis, how can you think about it using this?
You have to...
Just kind of think outside the box.
You have to think a little, you have to make the connection of that if you're not getting glucose in the cells, your brain is thinking that you're starving.
Yeah.
Okay? That's what you gotta think about.
Yeah, no.
Yeah, and then it fits into the ketogenesis.
That's why I keep going, like, is it high or low? But it's high blood sugar, but your body thinks you're starving.
Yes. And that's the confusing thing about it. It is a confusing thing, and you can't really, fit all of it in and try to reverse it because you still have some of this and some of this, right? So, but basically, overall, your body is going to be responding like it's starving, okay? And that's why, you will have some of this, some of these things as well. But really, all I really care about for this, without going crazy, is ketogenesis. Because that is a major problem and symptom of uncontrolled diabetics where you can actually smell it on the breath. Again, I think she talked about it. And
but why is that? It's making fuel for your brain, or what?
But it's acidic, and so it's going to acidify the
Oh,
blood.
okay.
So
So
you
it's
don't want
metabolic
high levels of
acidosis.
ketones in because it causes metabolic acidosis. So, again, you're starving, you're surviving a little longer and it's worth it. But again, for a diabetic, you want to get that under control or that becomes, again, all the problems of acidosis.
So what was the confusing thing about it, that your body thinks you're starving, but
You're up here, but we're talking about things happening like down here. Like you're hypoglycemic, and you actually are hyperglycemic. But again, it's you got to just forget about it for a second and just realize your body does, since your body reacts to this, okay? And if you're not getting glucose in the cells, it's assuming you don't have glucose, basically, right? And you're starving. So it's going to stimulate hunger, okay? You have high blood sugar, which on your list says, that means we're not going to be hungry. Even with type 2, you have high insulin, which means you're not going to be hungry. But you have this symptom of diabetes called polyphagia, but it's excessive hunger because your brain is all fooled by this. It thinks you're down here, even though you're up here, because you're not able to get the glucose in the cells. We have receptors for that. But it is really complicated.
Yeah.
So.
are you talking about type 2 or type 1?
You could be talking about both when it comes down to it, because in both cases you're going to have a high blood sugar, but it's not getting into the cells in both cases, and therefore your body is thinking you're starving.
So in both cases, you're both hyper.
Yeah, you're hyperglycemic, right. In both cases, you have high blood sugar. In one case, you don't have insulin. In one case, your cells aren't responding to insulin. Either way, blood sugar is remaining high. So I don't know if that answered some of it. It answered another question on when I was asking about some of this stuff on one of those extra questions.
You said, I think in one of the past lectures, we have to know about norepinephrine and epinephrine.
Oh, yeah, only, and really epinephrine, only, and in fact, I don't know if I have a question on that, but we should put it anyway, that when we did this, that cortisol, when you looked at the slide for it, it was necessary, the one with the bullet points, we put it over here, it was necessary to fold glucagon and catecholamine, which is epinephrine and norepinephrine, but I kind of just put epinephrine down here. So epinephrine, adrenaline, fight or flight type thing, your life's being threatened. Hypoglycemic means your brain is being threatened. So you are going to, again, you don't have to list all these, but epinephrine is sort of falling in this category of doing some of these same things. In fact, we talked about it, that that helps release glucose from the blood, the liver, right? To help us get energy, into it, right? It opens up the blood to it. So it's doing some of these things here. So the bottom line is you sort of fit cortisol and epinephrine on the bottom rung here. It's going to respond to the same things. Hypoglycemia, what's going to respond to it and react to it? Glucagon, cortisol, epinephrine are all going to be released under those conditions and do basically these processes to survival, to help bring it back up.
It's do all 5.
Well, I'm not going to say like epinephrine does all 5, but I'm saying in conjunction, these 3 will do these 5 things. So I'm not saying everyone does all 5. I'm not going to try to even
And we
figure
don't
that
have
out.
to differentiate what does what.
What do you mean? You have to,
We don't have to differentiate.
well,
specifically.
glucagon does all 5, but you're not going to have to know specifically what those do. Just know they're in that same process. That they respond to the same signal and they're in the same process. But for glucagon, those 5 definitely occur. You have to know that for that. The other ones, I'm not saying specifically what they do, but they're working in conjunction, doing the same thing. So what other questions are on those extra questions? What do we need here?
The local parenting control,
See, again, that's all this slide. So if you go back, this is 22.
Is it innovation of peristalsis and gastric acid secretion?
So it's all this.
Number 4.
So this is the slide you have to know to answer that question, which is barely much, at least I don't know the whole question, but as far as, because I have to look at it again if I ask any like little things. Well, I talked about inhibition and stuff, right? Remember for this chapter, and even for the first 2 chapters were pretty straightforward, right? What was happening at the stomach and the small intestine, and what was the hunger and satiation column, and this. And so they're pretty straightforward questions. I'll go over this again. But all I could do was talking about, to make it more complicated, inhibit, what would an antagonist to this do? Or, an agonist for leptin or something like that. That's the only way I could kind of make this complicated. And of course, I'll have a couple of those. But just again, when you look at this, the idea is we're going to have local control of this. So when food, again, and it really is nice when you look at this. I'm saying everything that's written here. It's actually really well written. That when food enters the small intestine, what we want to inhibit the upstream process, because it's going to take a while, and we want to stimulate the process that we talked about in the previous chapter of getting all these secretions from the pancreas and liver in and start digesting. And it's cool, it's under local control. So once the food enters, it's, which is the chyme, right? That's sort of what we're calling the partially processed food from the stomach. When it enters, that the small intestinal cells themselves, and I don't care about the specific type, are going to release those 2 hormones, secretin and CCK. Okay? And again, 4, 5, and 6 are all we're reading here. Both of them are going to inhibit the upstream process of gastrin and peristalsis, okay, that's trying to move food further down, because we want to hold up. That makes sense. Now we want to get the secretions. So CCK is doing the main work here. It's getting the bile in from the gallbladder. And what's the purpose of bile? Emulsify fats, okay? And it's getting in and I'm going to even put that slide up a second. It's getting all the enzymes, that are coming, which is trypsinogen and friends. I'll actually show that slide, that we need to do digestion from the pancreas. So that's what CCK is doing, besides being involved in hunger, by the way. So here it's local. They both are doing this. CCK is getting the bile in, and then it's getting in the trypsinogen and other zymogens. Secretin is getting the bicarb. We're going to produce bicarb in here, or we're going to secrete bicarb in order to neutralize that stomach acid. Okay, and that's what it does, and it's right there. It goes directly to the previous chapter. In fact, there's something else I want to talk about, endocrine exocrine. That will help you guys too. So sort of, well, this is sort of the the word you want to tell you what secretions we're getting here. But here again, when you look at sort of what's happening is, it's mainly happening right here. Food's entering. We have the other slide with bile coming in to help digest fats. But here's the idea of what CCK is getting in. It's getting in these pancreatic secretions, which are all these zymogens. And I call them trypsinogen friends because I don't want you to memorize all these lipases and carboxypeptidases, because it's going to be sort of a cascade where when it comes in, so this is what CCK is doing, getting these things in yellow in, so it can do it. Now you remember though from the previous chapter, the key is the small intestinal cell itself will activate this, convert it, the zymogen to trypsin, by something called enteropeptidase. And then this cascade happens. And because of this, we're going to start digesting peptides and fats. Okay? Remember, lipases digest fats, and the peptidases, including things like trypsin, will digest proteins. So that's the process here as far as that goes. But the key is, well, what's going to cause this to happen? Well, when food enters, it's going to help get this in. Okay? So CCK will actually cause all the secretions to come in, and enteropeptidase will then convert it and will start digesting. So it's a coordination of the events. So that slide kind of shows what's happening over here, okay, in the previous chapter. It's going to coordinate all this happening.
When it says trypsin activates other coenzymes, what are they? So trypsin activates the ones in yellow
Yes.
to turn into the ones in purple?
Yes.
Okay.
And those are the ones you don't need to know the names of, but like we talked about before, you got to realize those are going to digest proteins and fats. Okay, but you don't need to know the names. But you do need to know trypsin and associate with that small intestine. What zymogen/enzyme protease do you need to associate with the stomach? Pepsin and pepsinogen. So just to kind of review that too, for the stomach, you're looking kind of at the 3 cell types and what they do. That's the main thing from the stomach. The saliva, if you take this journey, again, which is kind of mirroring that, saliva, that's what you need to know. Amylase, which digests carbohydrates, and lipases digest fats. Boom. The stomach, you have to realize the secretions and what cell types do it. Okay? Parietal cells, hydrochloric acid, which will help the chief cells, pepsinogen. Okay? Hydrochloric acid converts mucus, mucus and bicarb. So you need to know that. And again, for the small intestine, this. Plus the idea of bile, which is this.
The enzyme that's coming from the stomach that enter and turn like the one that converts pepsinogen
To pepsin?
yes,
No. Trypsinogen?
trypsinogen into
Trypsin is coming from the small intestinal cells themselves.
oh,
So-
Today?
The enteropeptidase.
that's not coming from the stomach.
No.
It's from the small intestine.
This one. So right, because that's where what the action occurs. That's occurring right in the. Thanks for the food again.
Yep.
That's when food enters.